Hey guys, this is the fourth concept for Unit 2, the fourth and final concept. Unit 2 covers sections 5.3 through 5.5. This is the one that we went over in class. This is the voiceover for the PowerPoint presentation so that you can have a reference to go back and use. First thing are the types of cellular transport. Because the plasma membrane, that phospholipid bilayer, is semi-permeable, which means that it only allows certain substances to cross easily into and out of the cell. There must be a method for things to get in and out of the cell, and there's two ways that cell transport can occur. It's either going to be passive transport, which is what we see here, or it's going to be active transport. Now, the difference between the two has to deal with the concentration gradient, or the difference in particles across the membrane and the way in which they move. Whether or not there is an energy requirement is also one of the differences. So with passive transport, you're going to either have three modes. To passively go from high to low, a cell will use DFO, where D is diffusion, which represents simple diffusion. The F is for facilitated diffusion, where a protein must be used as a method of getting the substances across the membrane. And O represents osmosis, which is the movement of water. It is only the movement of water. All right, so passive transport, again, is simply crossing the plasma membrane. These are going to be things that are small and nonpolar. It must meet both requirements. It must be small and nonpolar, not or. And then facilitated diffusion, typically things that are too large or that carry a charge. So if they're too large or carry a charge, they will have to go through facilitated diffusion in a passive transport situation. Facilitated diffusion is going to use an integral protein, an integral transmembrane protein, meaning that that protein is going to span the width of the plasma membrane. And there's two types of integral proteins that do this. We have the channel proteins, which are always open, and we have carrier proteins. And carrier proteins have to actually make a conformational change in shape in order for substances to be passed across the membrane. The purpose of an integral protein is so that substances that are too large or charged can actually avoid the hydrophobic region of the plasma membrane, so it's a bypass way system. Both of these proteins are going to be specific for what they allow to pass through. So even like with the channel protein, even though it's an open channel, not every large or charged substance can pass through. So there is some type of specificity that goes along with these integral proteins. Active transport is the opposite of passive transport. With passive transport, substances are going to diffuse with the concentration gradient from an area of high concentration to low concentration, which is the natural tendency of particles. As they're moving around and bumping into each other, they naturally spread out. So from an area of high concentration to low concentration, and because this is the natural tendency of particles, there's no net energy requirement to get this done. Active transport is going to be the opposite. Active transport is what happens when substances are going to move against their concentration gradient. So the natural tendency would be to go, like if you look at these diamonds, the natural tendency would be to move from an area of high concentration to an area of low. However, the cell needs a high concentration on the inside. So it wants to keep the concentration on the inside higher than the concentration outside. So in order to do that, it requires movement from low to high, which is against the concentration gradient. And in order to move against the gradient, some type of net energy is going to have to be used in that process. Now, this is an example of a sodium-potassium pump, where the cell is attempting to keep the concentration of sodium low inside the cell and the concentration of potassium high inside the cell. The sodium-potassium pump is the most commonly used form of active transport for animals. In plants, fungus, and bacteria, we're going to see that most commonly used form is the proton pump, which is going to pump hydrogen ions across the membrane. That's an overview, and I'm probably going to say some of this stuff again as we go through the slides. So again, passive transport is diffusion of a substance across the membrane with no net energy investment, and it's based off of the tendency of molecules to spread out evenly into the available space. So molecules will randomly move from an area of high concentration to an area of low concentration, and this is going to most often exhibit what's known as directionality, which is that moving from high to low. Now, at dynamic equilibrium, because that's the tendency, is to try to reach a dynamic equilibrium where you have a set amount, where you have about an equal amount on both sides of that membrane. But at dynamic equilibrium, the movement of particles does not stop. It's just that the directionality and the rate is going to be equal. So they're going to move across the membrane, excuse me, across the membrane in both directions at an equal rate, which keeps the concentration gradient at about an equal state. That is the tendency of cells to try to reach a dynamic equilibrium. When you have the diffusion of solutes, remember in a solution you have a solvent and a solute. A solute is a thing that is being dissolved in the solvent. Now, typically we think of solutions as being a solid particle being dissolved in a liquid particle, which is most often true. And the liquid for the cells is typically water. So when you're looking at diffusion where you have either passive diffusion, simple diffusion, or facilitated diffusion, we're typically talking about the solutes, what's happening with the solutes, because the movement of the water is referred to as osmosis. So as the solutes cross that semi-permeable membrane, if it does so easily, without needing assistance, we call that simple diffusion. However, if it needs assistance going through a membrane protein, we call that facilitated diffusion. But again, the particles are going to spread out from an area of high concentration to an area of lower concentration in hopes of reaching dynamic equilibrium. Now, this can happen. We can look at it with one particle. We can look at it with multiple particles. The goal, again, will be for the particles to reach that dynamic equilibrium. And this is only true for the particles that can cross that membrane, either needing assistance or whether they can simply cross. Only particles that can cross the membrane can seek that dynamic equilibrium. Typically, that's going to be any of the particles, though, because the ones that can't just go through the membrane will receive assistance will use the assistance of a membrane protein channel. I apologize, guys. I do not feel good, so if I pause for a minute, try not to sneeze or whatever. I'm sure y'all don't need to know that, but my brain's not completely functioning at this moment. Sorry. All right. Again, osmosis is the movement of water. Facilitated diffusion, again, is going to use a integral protein. Those integral proteins can be either channel proteins, which are open, but they are specific for what they allow to pass through, or it can be a carrier protein. When a carrier protein, again, if you notice here in figure B, the carrier protein has to make a conformational change in shape to assist membranes. To assist particles across that membrane. But it's passive diffusion because there is no net energy requirement, and there is a movement of particles from an area of high concentration to an area of low concentration. Now, please remember that integral proteins can be used in both passive transport and active transport. What makes these proteins you see here, a part of the facilitated diffusion passive transport method is that they are moving particles from high concentration to low concentration and doing so without the input of energy. If these proteins were moving particles from low to high using energy, it would be a part of an active transport system. So just because a protein is involved does not mean that it is active transport. What classifies it as active or passive is the direction of the particles, which direction they're being moved in, and whether or not there is an energy requirement. Please don't forget that. So here we have a diagram showing the effects of osmosis. Now, what most people seem to think is that when we're talking about the movement of water and water reaching dynamic equilibrium, that we want the same amount of water on both sides. That is not true. It is not about the volume of water, as we see here, but it is about the molar concentration of water. Water is going to, anything that is hydrophilic and can be dissolved in water is going to form an association with water molecules, as we can see in the picture below. Now, that creates a concentration or a molarity, which is going to be the concentration of the free water particles to the water particles that are in association with those solutes, okay? So the goal of osmosis is to have an equal concentration on both sides. So you want to have an equal concentration on both sides of the free water particles. So water is going to move from an area of high concentration to an area of low concentration, but the concentration is of the free water particles, okay? So these are, if you look here, these are in association. So you have like 3 in association with the solute. Over here you have 1. You have a bunch of free water molecules over here and very few over here. So that means that there is more water, more free water particles on the left of this diagram than it is on the right. So water will diffuse faster to the right to try to reach dynamic equilibrium. Now, does that mean water will not move to the left? No. You will have some movement, but if you notice the difference in the size of the arrows, indicates that the water movement from the left to right is greater than water movement from right to left. So the goal is again to reach dynamic equilibrium. So that is how osmosis works. Osmosis is the movement of water. Typically, when you have a problem involving osmosis, you'll be given the concentration of the solute and expected to infer the concentration of the solvent, which the solvent is water. All right, now tonicity is the ability of a surrounding solution to cause a cell to gain or lose water. The solution can be one of 3 types of solutions. You can have an isotonic solution where there is no net gain of water. The concentration of the water is going to be the same on the inside as it is on the outside. And so you're going to have equilibrium style movement and equilateral movement. So you're going to have the same amount going in as coming out. So there's no net movement. There is water movement, but there's no net movement. A hypertonic solution is going to have a solute concentration greater on the inside than the outside. And we're talking about what the environment, the solution of the environment is doing to the cell at this point. So make sure that you pay attention to what you're being asked to determine, because if it's what the environment is doing to the cell, it's going to be the opposite of what the cell is doing to the environment, okay? So a hypertonic solution is going to cause the cell to lose water because the solute concentration is going to be greater inside the cell, which means that the water concentration is lower inside the cell than it is outside of the cell. So therefore, remember that water moves from an area of high concentration to an area of low concentration. So when the solution of the environment is hypertonic, it causes the cell to lose water. So the environment is hypertonic to the cell, but at the same time, the cell is being hypotonic to the environment. So keep that in mind. When one solution loses, the other is gaining. A hypotonic solution. So if the environment contains a hypotonic solution, it is going to have a solute concentration on the inside of the cell will be less than what's outside of the cell, which will cause, wait, what did I just say? Solute concentration is less in the solution than it is inside of the cell. So there's less solute in the cell than out of the cell, which means that the outside solution has more water. And so again, water moves from high to low. So the water will move from the environment to the cell. So the environment solution is hypotonic to the cell, but the cell is hypertonic to the environment. Okay. Now, the effects of tonicity, the effects of the way water is going to move, is going to be different for cells without walls than it is for cells with walls. So we know that there are some types of protists, such as paramecium, and all animal cells that do not have a cell wall. And for cells like paramecium, single cell organisms like paramecium, they must have a way to regulate the amount of water that is moving into and out of those cells, and that's called osmoregulation. For cells without cell walls, hypertonic and hypotonic environments are going to create osmotic problems for that organism. Paramecium, again, use what's called a contractile vacuole. Remember, a vacuole is a substance that can store, is an organelle that can store water. Well, with paramecium being a single cell organism, in order to control the flow of osmosis, so for osmoregulation, they use a contractile vacuole, which has the ability to pump out excess water when needed. And that prevents the cell from lysing, because for cells without cell walls, in a hypotonic environment, if water continues to pour into the cell, it can cause the cell to lyse or burst. In a hypertonic environment, it is going to pull water from the cell, which will cause the cell to shrivel. In both of these situations, the cell dies. The ideal solution for a cell without cell walls is an isotonic solution. Now, cells with cell walls react differently to the flow of water and use that cell wall for osmoregulation. Cells with cell walls are going to be some types of protists, your plant cells, your fungus cells, and your bacteria or your prokaryotic cells. In this situation, a cell with a cell wall, the best situation is going to be one where it is in a hypotonic solution, because this is going to create a turgid environment by increasing turgor pressure. And turgor pressure is that internal pressure exerted from water entering the cell. As water enters the cell, it is going to fill that central vacuole, which is going to put pressure on the internal plasma membrane, which will again put pressure on the cell wall, which will help this cell to become very firm. You can overcome the ability of the cell wall to resist the amount of turgor pressure, but it's hard to do. So yes, you can overwater a plant cell, but it's hard to do. So the ideal situation is going to be one where it is in a hypotonic solution. Being in a isotonic solution is one where there is not enough turgor pressure to actually have the cells be firm, so they're typically going to be limp. And what this does is like a droopy plant. And so this is the result of an isotonic environment. Now, in a hypertonic environment, this is really bad for the plant cell, because as the cell loses water, that central vacuole is going to be drained of the water as well. And because remember, water moves from an area of high concentration to an area of low concentration. Now, the danger in that is that the central vacuole is anchored to the cell with attachment to the plasma membrane. The plasma membrane is also attached to the cell wall. So as the central vacuole begins to shrink, it in turn pulls the cell membrane away from the cell wall. the cell begins to collapse and will eventually die. This is called plasmolysis. So once a cell becomes plasmolyzed, this is you actually have death that is starting to occur. Again, active transport is going to go against the concentration gradient. Active transport moves from an area of low concentration moves particles from an area of low concentration to an area of high concentration, with the expense of energy. Again, the most common active transport mechanism in animal cells is the sodium-potassium pump. Let's look at how that works. So a carrier protein is used with a sodium-potassium pump. And remember, carrier proteins have to make a conformational change in shape. When the carrier protein is open to the inside of the cytoplasmic side of the plasma membrane, it has an affinity or an attraction to sodium ions. The goal of the cell is to keep the sodium ions low on the inside of the cell and the potassium ions high on the inside of the cell. Remember, the natural tendency of things is to move from high to low. So the sodium natural tendency is to come into the cell, to go from high concentration to low. But we need to keep the sodium in the cell low. So we need to keep pumping out those ions as they get into the cell. Again, the natural tendency of things is to move from high to low. So the potassium ions, which is typically high on the cytoplasmic side, has a natural tendency to go out of the cell. But we don't want that. We want potassium in the cell, high amount of potassium in the cell. So we have to have a mechanism to keep bringing potassium back into the cell as it is moved out of the cell from high to low. So the way the sodium-potassium pump works is when that carrier protein is open to the cytoplasmic side of the membrane, it has an affinity for sodium ions. Now there's a 3-to-2 transfer. For every 3 sodium that goes out, 2 potassium come in. A student asked me, why was that? And if you think about the size of a sodium ion versus the size of potassium ion, there's a size difference there, which may account for that. I am not completely certain, and I must remember to look that up to find out though. But just using the chemistry knowledge, you would that may play a role in that. But moving forward, so once the sodium ions are bonded to the carrier protein, that carrier protein is phosphorylated at the expense of ATP. So remember, ATP is adenosine triphosphate. So there's 3 phosphate groups. Once ATP undergoes hydrolysis, one of the phosphate groups is removed, which causes a release of energy. That phosphate group is then, with the help of enzymes, of course, attached to the carrier protein. When this happens, this causes a conformational change in shape of that carrier protein. Now, when that carrier protein changes its shape, it closes to the cytoplasmic side, but opens to the extracellular side of the plasma membrane. By doing that, by changing its shape, it also changes its function. Remember, structure dictates function. So when it opens to the opposite side of the plasma membrane, it loses its affinity for or attraction to sodium. When it loses its affinity for sodium, the sodium ions are released outside of the cell. And at the same time that it loses its affinity for sodium, it gains an affinity for potassium ions. So the potassium ions are then attached to the carrier protein, which causes the phosphate to be released as inorganic phosphate into the cell, which is extremely important because it's going to be used later on to in the process of creating more ATP by bonding ADP with inorganic phosphate. But anyway, the potassium ions are going to bind to the carrier protein, which causes the phosphate to be released from the carrier protein. This is called dephosphorylation. Adding a phosphate is phosphorylation, removing one, dephosphorylation. And again, this is going to cause a conformational change in shape. So the carrier protein opens now to the cytoplasmic side, And guess what it does? It loses its affinity for potassium. So potassium is released into the cell, and now it has an affinity for the sodium ions all over again, which means this process can happen over and over and over, each time at the expense of an ATP molecule. Okay. Ion pumps are used to maintain membrane potential. And when we think about membrane potential, and this is going to become very important when we start talking about photosynthesis and cellular respiration, membrane potential is the voltage across a membrane. And the voltage is created by the concentration of ions. So how many cations, which are positive ions, versus the concentration of anions, which are negative ions, is created by a difference in the distribution of those ions across that membrane, which is going to create what's known as an electrochemical gradient. And the electrochemical gradient is the combined forces of the ion concentration and an electrical force, which is the effect of membrane potential on the ion's movement. So how are those ions going to be able to cross that membrane? So you have the electrical force, which is the movement of the ions across the membrane, which is in combination with the chemical force, which is the concentration of ions on either side of that membrane. So you're looking at, well, how many cations do I have versus anions on both sides, and how do they move across that membrane? That creates what's known as an electrochemical gradient. Well, the chemical gradient, again, is the concentration of the ions. So cations versus anions, that's the totality. What's the concentration on the inside of the cell versus the concentration on the outside of the cell? And then the electrical gradient is going to be determined by the force that's created as those membranes cross from one side, as those ions cross from one side of that membrane to the other. Membrane potential is often you're going to see in plants, in some animals, but it's more common in plants, in fungus, and in bacteria. That membrane potential is going to be affected by what's known as a proton pump. And a proton pump, remember, a proton is just a hydrogen ion. A proton pump is going to be able to pump protons or hydrogen ions across the plasma membrane. And when it does so at the expense of ATP, which is why this is a form of active transport. So it's going to be moving things from an area of low concentration to an area of high concentration. So I want more hydrogen ions to stay together, which is going to help control that membrane potential, because if I have a lot of anions on one side of the membrane, I want to keep a lot of cations on the other side of that membrane. The proton pump is called an electrogenic pump because it is helping to create voltage as it pumps those protons across the membrane. And what this is doing is helping to store energy. And because proton pumps are used.
Teachers, at this time, we need cheer and dance to be released for the pep rally. Cheer and dance and be released.
I know it was a pep rally. All right. So anyway, yeah, I forgot what I was saying. Okay, so it pumps the the ions across the membrane to help maintain membrane potential. Okay. Now, co-transport, on the other hand, is when transport across the membrane is coupled. And so what you have is the active transport of one substance is going to drive the fusion of another substance. Okay, so what we see here is where a proton pump is actually going to be driving the diffusion of hydrogen ions through a sucrose hydrogen ion or sucrose proton co-transporter. Now, the thing with co-transporters is that you have to have both substances moving across the membrane together. So the protons can't go without the sucrose, and the sucrose can't go without the protons. So what's happening with the proton pump? The proton pump is going to, this is the active transport mechanism. It is going to continue to pump protons from an area of low concentration to high concentration, which keeps a concentration gradient of high to low, right? And the natural tendency is to move from high to low. So these hydrogen ions or these protons are going to move back into the cell from the high concentration to the low concentration through the co-transporter. In doing so, they also help to carry sucrose across the membrane. So without a high concentration of protons being outside of the cell, sucrose will not be able to get into the cell. So you have to use active transport, or the proton pump's job is to keep the hydrogen ion concentration high outside of the cell to keep the gradient from reaching dynamic equilibrium. Because if the gradient reaches dynamic equilibrium, then sucrose would no longer be able to get into the cell. So by continuing to pump the protons out, you keep the membrane potential as such where the hydrogen ions need to get back in. Okay, I hope that makes sense. If not, we'll just ask a question in class and I'll try to explain it more next week. All right. The next thing is bulk transport. Bulk transport occurs across the membrane by exocytosis and endocytosis. Bulk transport does not require a membrane protein, but typically you're going to have bulk transport with large molecules such as polysaccharides and proteins. Remember, things that are large or charged cannot simply cross the membrane. Usually things that are not too big, polar or ionic, are going to go through membrane proteins. But what about those really, really big things that can't go through a membrane protein? They have to undergo bulk transport. Your bulk transport ends in cytosis. So you have endocytosis and exocytosis, where exocytosis is things leaving the cell, and endocytosis is going to be molecules or particles coming into the cell. There's three ways that endocytosis can happen: through the cell eating, cell drinking, or receptor mediated, which is very specific for what is being brought into the cell. Phagocytosis is cell eating. Penocytosis is cell drinking. And so the way that this works is going to involve the membrane actively forming a vesicle around the substance. So in penocytosis, which is right here, liquid-like particles are going, so the plasma membrane is going to form a vesicle around, so it kind of like buds around the particles, pulls them into the cell, into a vesicle that's going to be coated with proteins. With phagocytosis, same type situation happens, except the vesicle is not going to be protein coated, okay? It's going to be larger solutes. This is typically a large amount of smaller solutes, so they call it cell drinking. Receptor mediated, the plasma membrane is going to have these receptors on it, which these particles will have to be able to fit the receptor. And when it does, it triggers the plasma membrane to form this vesicle around it and bring it into the cell. Again, exocytosis is the opposite. In endocytosis, you're bringing it in. In exocytosis, you would already have the substance in a vesicle, and then the vesicle would fuse with the membrane and push it out. That's it. Hope you're ready for the test. Rock and roll.